Hey everybody, we're in the nervous system now. So what I want to do today is review a couple models of the neuron. And what's important to keep in mind is that the neuron is the structural and functional unit of the nervous system. The brain and the spinal cord, which compose the central nervous system, are made up of billions and billions of individual neurons. The peripheral nervous system is made up of sensory neurons and motor neurons that travel through spinal nerves and cranial nerves. And so it's important to understand the structure of these individual neurons. And this is what we're looking at right here. So let's just go ahead and review these parts. Let's start up here. And the first parts that I want to identify are these branches, these individual branches that I'm pointing out here. What are these? Those are what we call dendrites. And dendrites are the receptive regions of the neuron. And so they're going to be receiving information most of the time from other neurons. There's going to be neurotransmitters that are being released from other neurons, and they're going to act on these dendrites. And so the dendrites are receiving this incoming information. What about this big region right there that contains this structure, which is the nucleus? What do we call this whole area? That's the cell body. Okay, so we have the cell body. Then the cell body gives way to this kind of funnel structure right here that we call this is what we call the axon hillock. And the axon hillock gives way to this long, slender structure, which we can refer to as the actual nerve fiber. And this structure is the axon. And then the axon are surrounded by these fatty sheaths. Not all the time, but in a lot of neurons, they're surrounded by these fatty sheaths. And what do we call these fatty sheaths? Those are the nodes of those are myelin sheaths. So here's a myelin sheath. There's a myelin sheath. And then the gaps in between these myelin sheaths, this is one right here, but they're all along the neuron in between these myelin sheaths. These gaps are called the nodes of Ranvier. And then it's not shown here, but on the end of this axon, there would be something like this, which represents the axon terminal. And that axon terminal is going to release neurotransmitters that act on other neurons or onto effector organs. Okay, so let's review some of the functions of the neuron. Let's just talk about how they work. Like I mentioned, these are the dendrites, and the dendrites are receiving information from other neurons. There's other neurons that are releasing neurotransmitters onto these dendrites. And that's actually what these little Hershey drops here, these little Hershey drops with these long, slender projections, these actually represent neurons that are synapsing on this particular neuron. They're releasing neurotransmitters onto this neuron model. And there's hundreds, perhaps thousands of neurons that are acting on this neuron. Not only on the dendrites, primarily on the dendrites, but you can see they're also synapsing here on the cell body as well. And so these neurons are releasing neurotransmitters onto the dendrites. And these neurotransmitters can be either excitatory, they can excite this neuron, or they can inhibit it. And we'll just go with the excitatory neurotransmitter. And what happens is if an excitatory neurotransmitter is released onto the dendrite. It binds to a receptor on the dendrite that opens a channel, and that allows an excitatory ion, most of the time sodium, positively charged sodium. It allows sodium to rush into the neuron, into the dendrite. And something that we have to know with neurons is that the inside of the neuron, right underneath the plasma membrane, has a negative charge. And that's called the resting membrane potential. And it can be, we'll say minus 70, maybe minus 90 millivolts. Anyways, it has a negative charge. And so when these sodium channels open, sodium rushes into the neuron. One, it's flowing down its concentration gradient, it's moving by diffusion, but it's also moving down its electrical gradient as well. That positively charged sodium is being attracted to the negative charge right inside the neuron. And what happens is that positively charged sodium follows that negative charge all the way down to the axon hillock. And the axon hillock is essentially receiving all of this positively charged sodium. And if enough sodium reaches the axon hillock, if the resting membrane potential at the axon hillock rises up to a certain level, what we call the threshold level, then an action potential is going to be generated. And all of these voltage-gated sodium channels open, and a whole flood of sodium floods into the axon hillock, and it creates this, what we call a depolarization event. It creates an action potential where, again, all this sodium rushes into the cell. And what does that sodium do? Well, that sodium follows that negative charge down the axon, down underneath the myelin sheath, and then it gets to the node of Ranvier. And at the node of Ranvier, there is a whole bunch of voltage-gated sodium channels. And so when this positive sodium reaches the node of Ranvier, again, it causes the resting membrane potential to rise up to a threshold level. These voltage-gated sodium channels open, and sodium, again, rushes into the cell, and you have another action potential that's generated at the node of Ranvier. And then that sodium, again, travels down, and it stimulates an action potential at the next node of Ranvier. And it's going to do that until it gets all the way down to the axon terminal. And we'll talk about what happens when it gets there. So something I wanted to point out, when you have these myelin sheaths, the only place that action potentials are generated are at the nodes of Ranvier. You don't have new action potentials. You do not have new action potentials that are generated underneath these myelin sheaths. And you may ask, okay, well, why is that significant? Well, the reason that's significant is when you have myelin sheaths and you only have action potentials being generated at the nodes of Ranvier, that speeds up the transmission of that electrical message traveling down the axon. It speeds up the rate that that information is traveling down the axon. There are some neurons in the body, for instance, motor neurons of the autonomic nervous system, for instance, do not have, or generally do not have, these myelin sheaths. And what that means is that you have action potentials that are generated all the way down the axon. And in the relative nature of the neuron, that just takes longer. When you have action potentials that are generated all the way down the axon, it takes longer for that information to travel down to the axon terminal. And so myelin sheaths speed up the rate of communication because you only have action potentials that are generated at the node of Ranvier. Before I forget, we're looking at the layers here. This is the axon, this is the myelin sheath, and this outer layer represents what we call the, I'm going to pronounce this incorrectly, the epineurium. And the epineurium is basically just the outer connective tissue layer around the individual neurons. And it's connecting it to other structures around the neuron. It provides structure for capillaries to support the neuron and that type of stuff. All right, let's move on. So we have those action potentials that are generated all the way down the neuron until eventually they get to the axon terminal. And what happens when action potentials get at the axon terminal? Well, again, that positive sodium reaches the axon terminal, and what it does is it opens channels called voltage-gated calcium channels. So the positive sodium charge comes down here, it opens voltage-gated calcium channels, and calcium rushes in to the axon terminal. What happens next? Well, what are these little pearl-like structures here? What are these little fish egg looking guys? Those are what we call vesicles, and vesicle is just a generic term for a circular structure, a circular kind of compartment within the body. So these are vesicles, but what do we find inside those vesicles? That's where we find neurotransmitters. And what's going to happen is calcium rushes into the cell, and that causes these vesicles to fuse to the plasma membrane and release those neurotransmitters out into what we call the synaptic cleft. And so it causes neurotransmitters to be released. And where are some places that neurons can release neurotransmitters? Well, one, they can release them onto other neurons. And so you can release these neurotransmitters onto the dendrites of other neurons, and that can either excite the neuron or it can inhibit the neuron. What are some other places that this axon terminal can release neurotransmitters onto? Well, it can release it onto what we call effector organs. And what are the organs, what are the tissues that the nervous system has control over in the body? Well, one, it has control over muscle tissue. It has control over skeletal muscle, smooth muscle, cardiac muscle, and it also has control over glandular tissue. And so these neurotransmitters can be released onto any of those. Here's a question for you. If this neuron is being released onto, we'll say, a skeletal muscle, if this neuron is releasing neurotransmitters onto a skeletal muscle, what are the neurotransmitters that are going to be released? And the answer to that is acetylcholine. Acetylcholine is the neurotransmitter that's released onto skeletal muscle to stimulate contraction of the skeletal muscle. If we're dealing with smooth muscle, cardiac muscle, or glandular tissue, the neurons that are going to innervate those type of tissues are part of what we call the autonomic nervous system. So the autonomic nervous system has control over smooth muscle, cardiac muscle, and glandular tissue, and that's all involuntary. And the neurotransmitter that it releases onto those tissues just depends whether it's the parasympathetic division acting on those tissues or the sympathetic division acting on those tissues. If it's the sympathetic division, the fight or flight division, then the neurotransmitter that's going to be released onto those tissues is norepinephrine. If it's the parasympathetic nervous system, the neurotransmitters that are released onto those tissues is acetylcholine. And the last thing that I want to point out is that most of the neurons in the nervous system have this general structure where you have all of these dendrites that are connected to the cell body, and then you have one long axon that projects to an axon terminal. But not all neurons in the body look like that. And sensory neurons are an example where they look a little bit different. So here we have a model of the skin. This is the outer layer of the skin. Well, let me just put it this way. This area right here is what we call the epidermis. This is stratified squamous epithelium. And then below that, we have the dermis, which is connective tissue. It's areolar connective tissue and then dense irregular connective tissue. And then below that, we have the subcutaneous layer, which is mostly adipose tissue. Well, the stratified squamous epithelium, the epidermis, has these little projections that we call dermal papilla. Essentially, the dermis has these nipple-like projections. That's what papilla means, nipple-like, that project into the epidermis. And the reason I point this out is because in these dermal papilla regions, we have a whole host of sensory receptors. And a lot of these are touch receptors, different types of touch receptors. Light touch, heavy touch, pressure, vibration, all stuff like that. But you can also have thermoreceptors and nociceptors here as well. Sensory receptors have a little different structure than what we call these multipolar neurons here. And what happens with sensory neurons, and I wish I had a diagram of this, but essentially the sensory receptor region here is modified dendrites. So in sensory receptors, the dendritic region is modified, and that actually forms the sensory receptor region. And this is where it gets a little bit different, is you have this sensory receptor, the modified dendritic region, but the cell body is not located in that same area. So what you have is you have the sensory receptor, then you have this long axon that's going to travel all the way to the spinal cord. It only takes one sensory neuron to go from the skin on, we'll say, the arm, all the way to the spinal cord. So you have this long axon that travels through a spinal nerve, and it's going to go to the spinal cord. And right before it gets to the spinal cord, actually I have a model I can use for this. So this is the spinal cord. This is a spinal nerve. And so you have that sensory neuron that comes in through this spinal nerve. And then what's going to happen right before it gets to the spinal cord is it's going to branch. And right back here is what we call the dorsal roots. And all of the sensory neurons that come in are going to travel through this dorsal root. And they're going to synapse in the spinal cord, and they're going to give the spinal cord that sensory information. But there's this structure here called the dorsal root ganglia. And this is where the cell bodies of all of these sensory neurons are located. So you have the axon that comes in, and then it projects up. You have this little cell body, and then the axon continues on, and then the axon terminal is located in the spinal cord, and it's synapsing with neurons in the spinal cord. So a little bit different structure with the sensory neurons.